Good morning. Today I will discuss the Module 2, the chemical basis of heredity. Number 1. Differentiate DNA from RNA. DNA, deoxyribonucleic acid molecule that acts as the mechanism of biological inheritance in almost all living creatures. DNA is found in nearly all cells and contains the coded instructions that control the working of the cell. DNA is passed from parent to offspring and contains the coded instructions that enable the offspring to develop from a single cell into an adult body. Ang DNA o tinatawag na deoxyribonucleic acid acts as a mechanism of all living creature. Ang DNA ay matatagpuan sa lahat ng mga cell. Ang DNA ay ipinapasa mula sa magulang hanggang sa mga anak at na-contains nito ang code instruction upang bumuo mula sa isang cell. Function of DNA. DNA is functional is the transmission of genetic information. It forms as a media for long term. Ang function ng DNA ay siya yung nagtatransmission ng genetic information. Predominant structure of DNA. The DNA is double-stranded molecule that has a long chain of nucleotides. Ang predominant structure ng DNA is double-strand molecule. The DNA exists in unit called genes. To a crude approximation, one gene code for one protein. Most bodily functions are at molecular level, carried out by protein. Some proteins act to digest, other proteins act to defend our bodies from infectious disease and others transport materials around the body. A few thousand kinds of protein are needed in the working of one cell, and a few tens of thousands of proteins are needed in the working of multicellular bodies such as human body. These thousands of kinds of protein are coded for in the DNA by thousands of genes. Ang DNA is nag-exist siya sa protein na tumutulong sa ating katawan. Ang protein din yung tumutulong sa ating para makaiwas tayo sa mga infectious disease at iba pang nakahawang sakit. The difference between DNA and RNA. Sa DNA, may thymine siya, cytosine, adenine, and guanine. Ang base pair niya is yung thymine, yung kulay red. Then, lahat ng apat niya na tinatawag na nucleobases of DNA. Sa RNA naman, may uracil, may cytosine, may adenine, and may guanine. Tinatawag din siyang nucleobases of RNA. Difference between DNA and RNA. Cytosine siya yung kulay green. Sa yellow siya yung guanine, nucleobases. Adenine siya yung pink and uracil yung orange. Sa RNA naman, cytosine is green, guanine is yung yellow, adenine is pink, thymine is blue. Helix of sugar phosphate. Base pair niya yung cytosine and guanine. Ang nucleobases naman niya is yung guanine and cytosine. RNA, ribonucleic acid, is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. RNA and deoxyribose nucleic acid are nucleic acid along with lipids, protein, and carbohydrates. Nucleic acid constitutes one of the major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, RNA is found in nature as a single strand folded onto itself rather than a paired double strand. Yung RNA o tinatawag na ribonucleic acid is a polymeric molecule siya. At siya rin yung nagpo-form ng mga carbohydrates, nucleic acid for our life. Siya is single strand lang siya, like the DNA is double strand. Function of RNA. RNA is functional is the transmission of the genetic code that is necessary for the protein creation from the nucleus to the ribosome. Ang function ng RNA is transmission siya ng genetic code creation from the nucleus to the ribosome. Predominant of structure of RNA. The RNA is a single-stranded molecule which has a shorter chain of nucleotides. Ang RNA is single-stranded than DNA is double-stranded. Number 2 is differentiate transcription and translation. Transcription is the synthesis of the strand of messenger. Transfer or ribosomal RNA from a DNA template is called transcription. In the process of transcription, the sequence of nucleotides present on the DNA molecule is copied and carried to the cytoplasm by mRNA. The process of transcription takes place in nucleus. Ang transcription is isang strand ng messenger na nagka-transfer ng ribosomal to RNA from DNA. Pagkakasunod-sunod ng nucleotide na present sa DNA and carried from cytoplasm by mRNA. Translation. In the process of translation, the specific amino acids are picked up according to the codons brought by mRNA. The process of translation takes place in ribosome located in the cytoplasm. During translation, proteins are produced with the help of RNA. Translation is the process of translating primary transcript mRNA into a sequence of corresponding amino acid forming a peptide. Ang translation is the process siya ng takes place in ribosome na nakalocate sa cytoplasm. During translation, ang protein is nagpo-produce ng RNA, the process of translating primary transcript RNA. Number 3 is how do DNA replicate? The DNA is copied or replicated once per cell division. It is copied at every cell division within the lifetime of one organism and in the reproductive cell line that produces sperm and eggs. A large set of perhaps 50 or so enzyme catalysts see the DNA replication. DNA polymerases are the most important of this enzyme. Ang DNA copy ng every cell division within a lifetime of one organism. DNA replication is completed by the fact that the two strands of double helix are mirror images of each other. One strand can be thought of as going left to right across the page, but the other strand goes to right to left. A molecular level, this is due to the structure of deoxyribose in the DNA backbone. Deoxyribose contains 5 carbon atoms, which are conventionally numbered 1 to 5. The phosphate upstream is attached to carbon atom number 3, phosphate downstream to carbon atom 5. The E band in one strand of the double helix is opposite the pi band in the other strand. DNA replication niya is may 2-strand double helix siya. Deoxyribose contains siya ng 5 carbon atoms which are conventionally numbered 1 to 5. The phosphate downstream yung carbon atom sa double helix is opposite the band in the other strand. Summary of DNA replication. Number 1 is helicases unwind the parental double helix. Number 2 is single strand binding protein stabilize the unwound parental DNA. Number 3 is the leading strand is synthesized continuously in the 5'3" direction by DNA polymerase. Number 3 is after RNA primer is replaced by DNA by another DNA polymerase not shown, DNA ligase join the Okazaki fragment is the growing strand. Number 4, lagging strand is the synthesized discontinuously primary synthesis a short RNA primer which is extends by DNA polymerase to form a Okazaki fragment. DNA ligase and yung DNA polymerase yung parental DNA RNA Okazaki fragment being made. Siya yung napoproduce yung Okazaki fragment being made. Replicating DNA proteins at the replication fork, the helicase unwind duplicate DNA and single strand binding protein SSB coat at stabilize single-stranded DNA formed by strand separation is seen ahead of the fork removing superhelical tension caused by the strand separation. Note that the leading strand is synthesized continuously in the 5'3" direction, whereas the lagging strand is synthesized discontinuously as short fragments called Okazaki fragment. In general, DNA is replicated by uncoiling of the helix, strand separation by breaking of the hydrogen bond between the complementary strand, and synthesis of the two new strands by complementary base pairing. Number 4, discuss how a protein are made. Protein are made of hundreds of thousands of smaller units called amino acids which are attached to one another in long chain. The sequence of amino acids determine each protein unique, three-dimensional structure, and its specific function. The information to produce a protein is encoded in the cell's DNA. When a protein is produced, a copy of the DNA is made called mRNA, and this copy is transported to a ribosome. Ribosomes read the information in the mRNA and use that information to assemble amino acid into a protein. Ang protein ay binubuo ng mga maliliit na unit na tinatawag na amino acid. To produce protein is encoded in the cell's DNA. When a protein is produced, a copy of the DNA called mRNA at transport ito sa ribosomes. These are the structure of protein. Types of protein structure. Protein are biological polymers composed of amino acids. Amino acids linked together by peptide bonds form a polypeptide chain. One or more polypeptide chains twisted into a 3D shape form a protein. Proteins have complex shapes that include various folds, loops, and curves. Folding in protein happens spontaneously.